Hello, my name is Helen Anderson, and I'm a research associate here at the Allen Institute for Cell Science. Today, I'm going to give you a brief introduction into cell biology, specifically focusing on what is a cell, why they matter, and what we do with them here at the Allen Institute for Cell Science. So why do we care about cells? We care about cells because our bodies are made up of cells, skin cells, heart cells, muscle cells, and more. Every part of your body is ultimately made up of one or more different kinds of human cells. Because our bodies are made up of cells, if our cells get sick, so do we. So healthy cells mean a healthy body. Now, what is a cell? This is what you've seen in your textbooks. Many textbooks contain these images of brightly colored golf balls filled with neatly labeled structures. But what exactly is that? When we look under a microscope, we unfortunately don't see something quite as neat and pretty. Cells in real life are a little messier and a little harder to parse out. When we look under a microscope here at the Allen Institute, we rarely see just one cell isolated like you do in your textbooks, but instead see hundreds of cells growing together in these different clumps known as colonies. Now, I mentioned before a specific kind of cell that we use here at the Allen Institute, and that's a stem cell. Stem cells are one of the most common types of cells used in research, and they like to grow in these clumps known as colonies. The reason that we like to use stem cells in biology research is because they possess a certain quality known as pluripotency. In other words, stem cells are pluripotent. This means that stem cells are able to become or turn into many different kinds of cells. Cells in the human body that are important to human health. For example, I could take a stem cell and turn it into a liver cell or a kidney cell or a heart cell. Here at the Allen Institute for Cell Science, I work with our differentiation team. That means that I take our stem cells, which are pluripotent and able to become many different kinds of cells, and I differentiate or turn them into heart cells, also known as cardiomyocytes, which we can then study. Studies done with heart cells can be helpful for heart research, which can help us to cure heart disease, which is why this kind of research is important. As I mentioned, we use stem cells here at the Allen Institute for Cell Science, and stem cells like to grow in colonies. You can see in the image to the right that I've circled one of these colonies, but there are many different colonies in this image. If we zoom in on one of the colonies, we can even pick out individual cells within it. This is a close-up image of one of those colonies, and you can see that in red, yellow, and blue, I've outlined some of the individual cells that make up this colony. Now, why do cells like to grow in colonies? Stem cells like to grow in colonies because cells can influence and communicate with each other in the same way that you interact with your friends. You can think about colonies as cell neighborhoods where all of the cells are neighbors. When the cells are able to grow close together, they're able to form tight interactions and communicate with one another, which can promote their health or help the cells to stay healthier. So how do we go from what we see under the microscope in the image on the left, hundreds of cells forming multiple colonies, to the image that we see in our textbooks, one individual cell with neatly labeled organelles and structures? It takes a lot of work, and it takes the kind of work that we do here at the Allen Institute for Cell Science. So just like your body has organs, your cells have organelles. Those organelles are the structures that we see neatly drawn and labeled inside the individual annotated or labeled cell in your textbook images. But unfortunately, they're a lot harder to see in real life. At the Allen Institute for Cell Science, scientists use fluorescent tags to make these organelles easier to see. Now, I'll say this next bit a couple times because it can be a little confusing. As I mentioned, your body is made up of cells, and your cells contain organelles. In the same way that your body contains organs and the organs help your body to function, your cells contain organelles and those organelles help the cells to function. These organelles are made up of proteins. So we have proteins that make up organelles, organelles that help your cells to function, and cells that make up your body. Going backwards, your body is made up of cells which contain organelles, and organelles are made of proteins. Now, at the Allen Institute, we like to study organelles because organelles help your cells to function. So by studying the functions of organelles, we can study the function of cells. And cells matter because they make up your body. And so what happens to your cell can influence what happens to your body. At the Allen Institute, in order to make organelles easier to study, what we do is we use a special kind of protein called a fluorescent tag. Now, what do you think of when you hear the word fluorescent? I think of things that light up, that are bright, like flashlights and highlighters, like fluorescent yellow. That's because fluorescent tags work in much the same way as a highlighter does. So when we want to make a sentence stand out in a textbook, I might use a fluorescent yellow highlighter to highlight that sentence and make it stand out from the rest of the text. At the Allen Institute, what we do is we take a special kind of protein called a fluorescent tag and attach it to a protein that we're interested in. A protein that we're interested in is usually interesting because it localizes or becomes centralized at or congregates at, becomes heavily, very densely located at an organelle of interest. Then, when we look at cells that contain this tagged protein under a special kind of microscope, that microscope makes the tagged fluorescent protein light up so that we can see that tagged protein, which is now localizing in an organelle of interest within the cell. We can see it light up within the cell, and it makes it stand out in the cell under the microscope, so it's much easier to study. Regular microscopes use visible light that we can see with our eyes, but our special microscopes only capture these fluorescent tags. Here's an example that will hopefully make things a little more clear. I love a good visual, and I love this slide in particular because you can see how helpful it is to highlight an organelle of interest to make it stand out within the whole cell. So one of the organelles that we've tagged here at the Allen Institute is the nucleus. In the image on the left, I can kind of pick out the nuclei. Nuclei is the plural form for multiple nucleuses, but nuclei. Now, without tags, I can see the nuclei as kind of the darker gray spots within the individual cells, within the whole colony, but there's a lot going on and it's kind of messy and hard to see. Meanwhile, in the image on the right, we're looking at the tagged nuclei. So the scientists here at the Allen Institute went through the process that I described before. They thought, I'm interested in cells, and I'm specifically interested in this one organelle within the cell, the nucleus. And I know that within the nucleus, there's this one kind of protein. So what I'm going to do is I'm going to take a fluorescent tag, which lights up when I look at it through my special microscope. I'm going to take a fluorescent tag. I'm going to add it to this protein. This protein is then going to localize or become located at the nucleus, the organelle that I'm interested in. Then I'm going to look at the cells that contain these fluorescent tags under my special microscope. And my special microscope is going to excite or make bright and make light up this fluorescent tag. And because the fluorescent tag is located at the nucleus, I'm going to see just the nucleus highlighted within the cell in the same way that you would see a piece of text, a line of text highlighted in your textbook if you had highlighted just that line. And that's what we're seeing here in this image to the right. This is an image taken through a special microscope showing just the nuclei that have been tagged with a fluorescent protein. As you can see, the tagged organelles are much easier to see and study than the untagged cells. At the Allen Institute, we've tagged many organelles and many different structures, not just the nucleus. In the upper left corner, you can see another structure in the cell that we've tagged, the nuclear envelope. The way that we did this is we took a fluorescent protein, one of those special kinds of proteins that lights up when we excite it by looking at it through the special microscope. And we took that fluorescent tag and we attached it to a protein called lamin B1, which you can see in parentheses there. The reason that we attached it to lamin B1 is that lamin B1 likes to localize or move to and become located at the nuclear envelope, the structure that we want to study within the cell. Then we took the cells that contained this tagged protein and we looked at them under a microscope. Our special microscope excited or made bright that fluorescent tag that was attached to lamin B1. And because lamin B1 is located at the nuclear envelope, then we see the nuclear envelope light up under the special microscope, which is what you see captured in the image in the upper left corner there. Now, why do we even care about seeing organelles? Yes, this is interesting. And we know that we care about cells because our bodies are made of cells. And we know that we care about organelles because they're in our cells. But why really do the organelles matter? It's not just about pretty pictures for your textbook, it's about cell health. Just like your body needs its organs to function for you to be healthy, a cell needs its organelles to work for it to function properly. In the same way that if your heart gets sick, so do you. If a cell's organelles get sick, so does the cell. You need your liver, your kidney, and your lungs to all work to feel good. And your cell needs its mitochondria, nuclei, and centrioles to function well for it to be healthy and function properly. Now, who uses these tagged cells? Once we've made a tagged line and we've taken our pretty pictures, what do we do with it next? Well, students of all ages can use tagged cells to study. It's helpful to look at the pictures in a textbook that have been hand drawn, at the illustrations that we saw before, the beautiful orange golf ball filled with neatly labeled structures. But it's also really helpful to see images of real live cells. Scientists at the Allen Institute also use the cells for research after we've tagged them. And other cell biologists also use these tagged cells for research. In the same way that doctors study the human body, cell scientists study human cells. And there are even specialists in cell science in the same way that there are in medicine. While a cardiologist is a doctor who specializes in the human heart, there are scientists that specialize in certain organelles. So if I'm a cardiologist, I study human medicine, but I'm a heart specialist. Some kinds of cell biologists are cell scientists, but they might specialize in a specific kind of organelle, like a nucleus specialist or a mitochondrial specialist. Being able to study an organelle with a tag helps that organelle stand out in the cell and therefore makes it much easier to study because you're able to see it more easily. So specialists in cell biology really appreciate these tagged lines that highlight their organelle of interest. Now, at the Allen Institute, we specifically like to study healthy human cells. Why study healthy cells? They're already healthy. What's the point in studying them? Well, it's to give us a baseline against which to compare sick cells. We need to know what cells look like when they're healthy so we know what they look like when they're sick. If we don't know what a healthy cell looks like, how would we know when it gets sick? And once we know that a cell is sick, we can try to figure out how to make it better. But first, we need to know what healthy cells look like so we know what unhealthy cells look like. And then we can try and make the healthy cells better. Remember, our bodies are made up of cells. So if they get sick, so do we. Healthy cells mean a healthy body, mean a healthy you. Here's an example of a disease in a cell that can then create a greater disease in the entire human body. It's a mouthful, but familial hypertrophic cardiomyopathy is a kind of heart disease. For some people, a mutation in their heart cells or cardiomyocytes makes their heart muscle too thick, and this thickening can impede healthy blood flow and normal heart function and can be life-threatening. Scientists are trying to fix the mutation in these people's cells to help heal their hearts and their bodies. Problems with your cells can create problems for your whole body, as familial hypertrophic cardiomyopathy is a great example of. Familial hypertrophic cardiomyopathy is just one example of a disease in which a problem with your cells can create problems for your whole body. Another kind of disease where a mutation in your cells can create a greater problem for your entire body is called progeria. For individuals with progeria, there is a mutation in their cells, specifically in a gene called lamin A, that makes their cells die prematurely. This causes their bodies to appear to age rapidly, and it can be life-threatening. Scientists are trying to fix the mutation in these children's cells to help heal their bodies. Now, what do you think a healthy cell looks like? We've seen some images so far of healthy cells under a microscope. Here at the Allen Institute, our healthy stem cells have smooth colony middles, rounded edges, and well-packed colonies. What that means is that when our cells are healthy in the lab, they have smooth colony middles because we grow them on these plastic plates in a liquid medium. Medium is a fancy word for liquid that contains the nutrients that cells need to be healthy. When our cells are healthy, they have smooth colony middles, meaning that the colonies or clumps that they grow in are nice and smooth because all of the cells are well connected to each other. They're holding hands very tightly. They're well packed. And they're also sticking down on the surface of that plastic plate that we grow them on very tightly. The colonies will also have smooth edges with rounded edges. Now, what do you think an unhealthy cell looks like? I know that when I'm sick, I look different than when I'm feeling good. When I'm sick, I might have a runny nose or a cough. I might look tired. Whereas when I'm healthy, hopefully I don't look the same way. Unhealthy cells also look different from healthy cells. At the Allen Institute for Cell Science, when our cells are unhealthy, the colonies may have spiky edges and the cells may look balled up. This balled up effect comes from, as I mentioned, we grow our cells at the Allen Institute on plastic in a liquid that contains their nutrients. When the cells are healthy and happy, they stay firmly pressed onto the plastic plate. But if they're unhealthy, they might start to pull up off of the plate a little bit, which gives them this balled up look. As well, when cells are unhealthy, sometimes those tight, close interactions with their neighbors become a little looser. They stop holding hands quite so tightly. And this can create empty spaces, gaps, or craters in the colonies, which you see imaged in that middle image there. As well, colonies of unhealthy cells may appear enlarged or flattened. Now, what makes a cell unhealthy? Cells can appear unhealthy for a variety of reasons. A mutation in the cell, like those discussed earlier when we talked about cardiomyopathies and progeria, can affect cell health. As well, some environments can make the cells sick too. I mentioned that we grow our cells in a liquid called media that contains the nutrients that cells need to be healthy. It also has to be at the right temperature. And cells can get sick if they get too cold or too hot, or they don't have enough or the right nutrients. Also, sometimes cells can look unhealthy if they're very old or very young. How do you feel when you get sick? Do you act differently when you're healthy versus unhealthy? I know that when I'm healthy, I tend to want to do things like go on hikes or go for a run. But when I'm sick, I usually just want to lay on the couch. Just like people, cells look different when they're sick or unhealthy, but they also act very differently. Healthy cells tend to hold tight to their neighbors. As I mentioned, they're holding hands very closely. They stay firmly grounded or they stay well connected to that plastic surface that we grow them on. They also grow quickly, grow big and strong. They divide, which is the way that cells reproduce. And they also move around. An unhealthy cell might grow and move more slowly, may detach from the plate, that surface that we grow the cells on, and may detach from its neighbors, causing that balled up look, causing the cells to become more isolated. And ultimately, unhealthy cells may die. In summary, cells look and act differently if they are healthy or unhealthy. Healthy cell colonies at the Allen Institute have smooth edges and form tight colonies. The cells move around and grow quickly. They may divide or reproduce, and they usually stay grounded to the cell plate, i.e., remaining attached to the surface that we grow them on in the lab, nice and flat, nice and strongly connected. Meanwhile, unhealthy cells may have colonies with spiky edges, and the cells may look balled up as they come detached from the plate. They may also detach from one another, move sluggishly, and ultimately unhealthy cells may die. So, what's the big picture? We've talked about a lot of things today, but ultimately, human cells are a lot like human bodies. Cells have organelles that help them to function properly in the same way that humans have organs. And these organelles can be hard to see, so we tag them with fluorescent proteins to make them stand out. I'll repeat that last step one more time because it's a little tricky. But we have bodies made of cells, which contain organelles, which are made of proteins. At the Allen Institute, we make organelles easier to study by tagging proteins with a fluorescent protein that lights up under a special microscope. That fluorescently tagged protein then localizes or becomes located at the organelle of interest. When we look at the cells that contain that tagged protein under our special microscope, we see the organelle, which is filled with all of those tagged proteins, light up or become highlighted within the cell under the special microscope, making it easier to study that organelle. As well, cells look and act differently when they're healthy or sick, just like you look and act differently when you get sick. At the Allen Institute, scientists study healthy cells so that we can identify sick cells, the cells that don't look or act healthy, and help those sick cells to get better. Cells matter because our bodies are made up of cells, and healthy cells mean healthy bodies, mean a healthy you. Now, let's say you're still interested in cell biology. There's a lot to learn out there. So, where can you go to find more information? You can start at the Allen Cell Explorer at allencell.org. Here we have two very cool resources about cell biology: the Integrated Mitotic Stem Cell and the Visual Guide to Human Cells, as well as many other educational outreach resources. There's also the Pacific Science Center's virtual programs, as well as the Genetic Science Learning Center, NOVA, and iBiology websites, which are all great starting points for cell biology research. Finally, I want to say thank you to the Allen Institute's founder, Paul G. Allen, for his vision, encouragement, and support, as well as a huge thank you to all of my colleagues here at the Allen Institute for Cell Science. And most importantly, a huge thank you to you for listening, for watching, and for caring about cell biology. Thank you, and have a good day.